So, hello everyone. In this demo session, I will show you how to design primers for a gene of interest. And we will do this using the NCBI website. So you can go to NCBI, you can open the website, and then here, instead of all databases, you will select gene. And then in this search box, you can search for your gene of interest. So in this example today, we will develop primers for the p53 gene, so a tumor suppressor gene from human. So what we will do is we will search here p53, and then you can immediately add the organism. So in this case, Homo sapiens. And then you press search. And then you will get this whole list of search results. And you will see that the one here at the top is already the correct one. And otherwise, you can also search further through the list. But I will click this one here. So then it will open the page with all the information regarding the p53 protein from human. So we have this over here. And then you can see on this page that you will find a lot of information on this gene. So here you can see the structure of the gene. And then when you scroll through to the bottom, you can see the expression of the gene in different human organs and tissues. You can find some publications on the gene and so on. But what we are interested in is finding the mRNA sequence of the transcript derived from this gene. So I will just scroll down a little bit more. But as you can see, there is quite a lot of information to be found on this page. So in case you cannot directly find the mRNA, you can also, of course, use the search function. So you can click Ctrl F on your keyboard and search for either mRNA, or you could also search for capital letters NM, because that's how the mRNA is denoted on NCBI. So I found the mRNA here. So the mRNA in this case is named NM000546.6. And this is the mRNA of the p53 gene. And this is then translated into a protein, and the protein is denoted by NP. But we are investigating gene expression levels, which is, of course, mRNA level. So we need to click here the mRNA. So you can click this. And then you will be directed to the page related to the transcript of this gene. In this case, it's transcript variant 1. As you could see on the previous page, there are several transcript variants of the gene. In general, when you want to measure the expression of a gene, you're interested in the expression of all the transcript variants. But of course, if you're interested in checking the expression of a specific transcript variant, then of course you should go to the page corresponding to that transcript variant. Then again, you will find a lot of information on the mRNA on this page here. And then from this part here, the features, you will find a lot of different features of the mRNA. So you will see the different exons that the mRNA consists of. And you will also find the so-called CDS. And CDS stands for the coding sequence. And this is basically the mRNA sequence without the UTR regions. UTR stands for untranslated region. So this part of the mRNA is not coding for a protein. The rest of the sequence is, so that's why we call it the coding sequence. So it's the part of the mRNA that codes for a protein. And we will generate primers specifically for this part of the mRNA. So therefore, we will click CDS, and then you will see here at the bottom of the screen or of this page, you find the entire mRNA sequence of the first transcript variant of P53, but now the coding sequence has been highlighted in this brown color. And you can make sure that this is the coding sequence, as it will start with the start codon, which is, of course, ATG. So ATG is always the start codon. So these should be the first three nucleotides in the coding sequence. In order to design primers, we should then copy and paste the sequence into a Word document. However, as you can see, it's not very convenient to do it in this way, because here there are several spaces between the different nucleotides, and you will also see that there is nucleotide numbers at the beginning of each line, which is not very convenient when you want to design primers. So what we will do instead is we will go to FASTA here at the bottom. And then when you click this, you will obtain this sequence in FASTA format. And you can see that this is, again, only the coding sequence, as it starts with the start codon ATG. So this sequence you can copy entirely into a Word document. So I will just press Ctrl-C. I will open my Word program, open a blank document, and I will then paste the sequence into this Word document. I will just put the name of the gene and the organism at the top of the page, because of course when you're designing primers for multiple genes, it's important that you still know at the end which sequence corresponds to which gene. So now I copied the sequence to this Word document, and even though you cannot see it, the sequence will still contain spaces, and these spaces are localized at the end of each line. And these should be removed, because otherwise we will run into some issues later on when we have to search for certain smaller sequences within this larger sequence. And of course, you could remove the spaces manually, but especially for longer genes, this would take a lot of time. So what we can do instead is select the entire sequence, then use the key combination Ctrl-H. So this is the combination that you would use to replace certain pieces of text in a document by something else. And in this box for behind find what, you should type this little sign here, followed by a P. And in the replace with box, you don't fill in anything. So this combination here will refer to spaces, and you will not replace them with anything. So this basically means that all spaces will be removed. So what you do is you click replace all, and then you see that my Word software indicates that it made 17 replacements in my selection. So this means that it removed 17 spaces. Then it asks whether I want to search the rest of the document, and then I indicate no, because otherwise it will also remove the space behind the title. And then I close this box. So now I have a consecutive sequence containing all the nucleotides corresponding to the first transcript variant of P53. Now, what you might remember from the lecture on primer design is that generally we try to design primers in such a way that they bind either on top of an exon-exon boundary or across an exon-exon boundary. So that would mean one primer binds in, for example, the first exon, second primer binds in the consequent subsequent exon, so exon 2. Of course, if we want to do this, we have to know where the exon-exon boundaries are located. And we can do this by going back to the mRNA sequence. So we will return to the page for transcript variant 1. And what we will do now is in this features list, we will look for the exons. The first exon that I find, I can click. And then you will see that here it highlights this, the sequence corresponding to exon 1. However, this part of the sequence is not part of the coding sequence because the coding sequence only starts further on. So this part of the sequence I will not be able to find in my Word document because there I only copied the coding sequence. So instead, I will have to search already for exon 2. And I can do that by either going again to the features list, searching there for the second exon, but to make things more convenient, you will see that in the NCBI window at the bottom, you will also have this tool where you can nicely go to the next exon. So you just click this arrow here at the bottom. And now you will see that the second exon is highlighted. And what you will do then, you will see that here the start codon ATG is localized here. So part of the second exon is still in the untranslated region, but the last part of this exon is actually part of the coding sequence. So what I will do now is I will copy the last few nucleotides of the second exon. I will copy this sequence, so I'll press Ctrl-C. And then in my Word document, I will press Ctrl-F to find the sequence in my document. I will then press Ctrl-V to paste the sequence that I just copied. And then you will see that the sequence is highlighted here in my document. So this was the last part of the second exon. So this means that this T nucleotide here is the last nucleotide of the second exon. And of course, this means that the subsequent A is the first nucleotide of the next exon, so exon number 3. So what I will do is I will select the T as well as the A, and I will highlight them. So this means that the first exon-exon boundary within the coding sequence is localized here between this T and the A. So this is the first exon-exon boundary within the coding sequence. Then we will look for the next exon-exon boundary. So I will click here in the NCBI page on the arrow to go to the next exon-exon boundary. And then I will again copy the last part of the sequence. I will search for this in my Word document, and then I will again highlight the last letter of the sequence and the first letter behind it, because the boundary between the two exons will be located, in this case here, between the G and the T. And I will then continue doing this for all the exons in the list. So I will now search for exon number 4. That one ends with the G here. So here the boundary is again between a G and a T. Here in this case, I will select this entire piece of sequence. When I paste this in the search window of Word, I will of course have to remove the space in the middle, and then it will be able to find it. So then I will highlight these two G's. When doing this, it's of course important to always select a sufficiently long part of a sequence, because in this case, if I would only search for RT-TG sequence, I would find that several times within my Word document. And then I don't know which ones of these would indeed be the last nucleotides of exon 5. So always copy a sequence that's sufficiently long to make sure that it only occurs once in the coding sequence that you have in the Word document. Let me go for exon number 6. I will copy it here, remove the space in between, and then I will find it here. And we do the same thing for exon 7. I found this twice. So then I will just double check. I think it will be this one. So here the letters in front of it are GA, whereas here it is CA. But on this page, it's indeed GA. So this means that this was the correct sequence. So that the next exon boundary is located here. And then we look for exon 8. Again, remove the space. And then we found the next boundary. Exon number 9. Is located here. And this is the end of exon number 10. And then exon number 11, we will not be able to find completely in this coding sequence because the last part of that exon is in the untranslated region. So therefore, we have now found all the exon-exon boundaries within the coding sequence. We can count them. So there is 1, 2, 3, 4, 5, 6, 7, 8, 9 exon-exon boundaries. So then I will add title called exon-exon boundaries. Because what we want to do now is make a list of the locations of these boundaries. So I know that the first boundary is located between this T and A in this highlighted sequence, but I now want to know if we start counting from the first A and the start codon until this boundary, at what location we find ourselves. So how many nucleotides precede this boundary? And I can do that by selecting the sequence starting immediately from the first nucleotide, and I select the entire sequence until the middle of this first exon-exon boundary. And then I click here at the bottom of my Word page, the word count tool. And then it will tell me that in my selection, there are 74 characters. So this means that the first exon-exon boundary is located at nucleotide 74. Then I will do the same thing for all the other exon-exon boundaries. And of course, every time you have to start selecting the sequence, starting from the start codon, including the start codon also. Again, select, in this case, until the middle of the second exon-exon boundary, press the word count option, and you will find that this second boundary is located after nucleotide 96. I will do the same thing for the third boundary. This is located at nucleotide 375. The fourth one is located at nucleotide 559. The next one at nucleotide 672. This one I did already, so the next one is this one. It's at 782. Then there is one at 919. And this one is at 993. And the last one is at nucleotide 1100. So now I have a list of 1, 2, 3, 4, 5, 6, 7, 8, 9 exon-exon boundaries. So that's correct. This means I didn't miss any. What we want to do next is to design primers in such a way that they're located either on top of an exon-exon boundary or around an exon-exon boundary. And we will do this using the primer tree plus tool. So I will go to the primer tree plus website. You will find online that there are different versions of this primer tree plus, but it doesn't really matter which one you would use, because basically they will all work in the same way. So when you just search in Google for primer tree or primer tree plus, you will find something like this. So this is the page. And what you will do then is you will paste here the source sequence. And the source sequence is the entire coding sequence, because we want to design primers anywhere in this coding sequence. So we copy the sequence and paste it into this box. Subsequently, most of these boxes we can leave empty. However, here there are some boxes which we will fill in. So for the primer size, as we discussed during the lecture on primer design, it's important to design primers with a certain size or within a certain size range. So we will select 18 as the minimum length for the primer, 20 as the optimum length, and 25 as the maximum length. For the primer TM, which is the primer melting temperature, we will use 59 degrees as the minimum, 60 degrees as the optimum, and 61 degrees as the maximum. For the primer GC content, this is something we will adjust as well. We set 40 as a minimum, 50 as the optimum, and 60 as the maximum. And then for the product size range, we will indicate 90 to 150. So this means the amplicon, so this the product that we want to amplify, should have a size between 90 and 150 base pairs. These are the standard settings we use in our lab, but of course you can adjust these based on your own experimental setup. And then one thing that I will still adjust is this part here, which asks for the number to return. This is the number of primer pairs that the primer tree software will generate for you. So this means when you fill in 5, even though there might be 100 good primer pairs, it will only show you the best 5 possibilities, or it will only show you 5 possibilities. However, it's possible that within these 5 possibilities, none of them bind around an exon-exon boundary. And then you would have to perform your search again. So therefore, what I usually do is I change this into a larger number, and I generally fill in 100. So this means that the software will generate 100 primer pairs. Of course, it's possible that it's not possible for the software to find 100 good primer pairs. So it will then just give you whatever number is possible. So if there is only 20 possible primer pairs that would meet the requirements that you filled in here, yeah, then it will only show you 20. But if you would leave 5 here, then even though there would be 20 good primer pairs, it would only show 5. The only downside of filling in a larger number here is that the search or the calculations of the software to generate the primers will take longer. But yeah, I generally do it in this way. You will see that depending on the version of primer tree you use, the page will look slightly different, but in general, yeah, the different things that you can fill in will be the same. So you click here, pick primers. The response was very fast. And then what you will see is the primer tree output. The output is, of course, always shown in pairs, because in order to do a qPCR or any PCR reaction, you need 2 primers, a left primer and a right primer, or you can also name it a forward and a reverse primer. So one of the 2 primers will bind to the forward strand of the DNA, the other one will bind to the reverse strand. So for the first primer pair, it gives you an extended result, let's say. So it will show you the left primer, and then for this left primer, it will indicate the start location, so at which nucleotide in the sequence does the primer start. It gives you the length of the primer, the melting temperature of the primer, the GC content of the primer, and then it also shows you these any values, so any TH, 3' TH, and hairpin. And these values tell you something about the self-complementarity of the primers. So how large is the likelihood that the primers will bind to each other, form primer dimers, or form these hairpin structures that we discussed. So these values, ideally, you want them, so the NETH, the 3' TH, and the hairpin, you want them to be as low as possible. So ideally, they would be 0. The other parameters, you can just double check to make sure, but normally these will meet the requirements that we filled in. So for example, the length of the primers will be between 18 and 25 base pairs, the melting temperature between 59 and 61 degrees, and the GC content between 40 and 60%. And then here at the end, you will find the sequence of the primers. And here it will also indicate the product size. So here you can see the size of the amplicon, it's 116, so that's also nicely in between 90 and 150 base pairs. And then for the first primer pair in the list, It will also show you the entire coding sequence that you copied into the primer tree page, and it will indicate using these arrows where the primers will bind. But of course, in this sequence here, we cannot see where the exon-exon boundaries are. So what we will do is for different primer pairs, we will check whether or not they bind around an exon-exon boundary. And we can do this by checking the locations where the primers bind and comparing them to the list of exon-exon boundaries that we just generated in Word. So here, for example, one primer binds at nucleotide 845, and the other one binds at nucleotide 960. So in this case, you can see that these primers would bind around an exon-exon boundary, because the left primer binds at nucleotide 845, and 845 is smaller than 919. So the left primer binds before this exon-exon boundary. The right primer then binds at nucleotide 960. And this value is larger than 919, so it binds after this exon-exon boundary. So the left primer binds before the boundary, the right primer binds after the boundary. So this means that they bind around an exon-exon boundary. And this is something we would like for the primers, because this reduces the likelihood that these primers would also be able to amplify genomic DNA. The primers would still be able to bind to genomic DNA. However, in genomic DNA, there would be an intron in between the two exons where each of the primers bind. And because this intron likely is rather long, the amplicon size would become so long that in a qPCR cycle, there is insufficient time to amplify a product from genomic DNA. Let's say the intron in between is relatively short, then it might still be possible that genomic DNA would also be amplified, but then you would be able to see this in this dissociation curve, as this would yield an additional peak in the dissociation curve at a higher melting temperature, because a longer product has a higher melting temperature. So based on the location of the primers, seems like they do bind around an exon-exon boundary. The other values were also quite okay. This NETH is 10.83. Ideally, it would be a bit lower, but yeah, this is something you could check also for other primers in the list. So you can check here. You can also find other primers that might be suitable. So here, for example, this primer pair number, primer pair number 2 binds at, the left primer binds at nucleotide 806, the right one at 945. So this primer pair would also be okay, because the 806 is lower than 919. 945 is higher. And here you can see that the any and hairpin values are all 0. And also the any values, or the 3' values that you find here at the bottom, are also 0. So these primers have a lower likelihood of forming primer dimers than the primer pair that we just looked at. So this is a primer pair that would be interesting to consider. What we can do then is verify in our Word document whether indeed this primer pair would bind around an exon-exon boundary. So to do so, we can copy the sequence of the left primer. We press Ctrl F and paste the sequence over here. And then we can underline the sequence and maybe also mark it in green. So this would be the binding location of the left primer. So the left primer will bind here. Of course, it will not bind to this strand, but it will bind to the complement strand. because there you will find the complementary sequence. Then for the right primer, we will not be able to find the sequence within our Word document, because it will bind to the forward strand, and hence we need to take the reverse complement of the sequence. You could do this manually, but the easiest thing to generate a reverse complement is just to use an online tool. So if you search for reverse complement on Google, you can you can just use any of the pages that you find here. Here you will copy the sequence of the right primer. At the bottom here, make sure that you select reverse complement and then click submit. And then this sequence you can copy and you can search for it in your Word document. And then indeed you will find the sequence here. So this is the binding location of the two primers. So the left primer binds here, the right primer binds here, and you can see that they bind around an exon-exon boundary. So the left primer binds before this boundary, the right primer behind the boundary. So based on binding location, these would already be good primers. However, we also need to make sure that the primers are specific for our gene of interest. So this means that the primers would only bind to p53 and that they would not be able to bind to other sequences within the human genome. And to check that, we can use BLAST. So we will again go to NCBI, but we will now type NCBI BLAST. Now we click to go to the BLAST tool. Of course, we need to use nucleotide BLAST because we're not working with proteins, we're working with nucleotides. And what we will then do is we will first BLAST the left primer. We will copy it here. Then we will go to genomic plus transcript databases. And then you can choose either between human genomic and transcript or mouse genomic and transcript. So in this case, of course, we have to select human because we developed primers for the human p53 gene. All the other parameters you do not have to adjust, but you will just simply click BLAST. Then you might have to wait for a little bit. And then at some point, we will find the BLAST results. And here you can indeed see the obtained results. And the gene or the transcript that the primers are most likely to bind to will be found at the top. And the more you go down in the list, the smaller the likelihood that the primers will bind to this transcript or gene. So what you will then find are different values. So you have, for example, the query coverage. So this means which part of the primer overlaps or is complementary to this specific gene. In this case, this is 100%. And this is also what you would expect because the primer was specifically designed to bind to this gene. The percentage identity tells you how many or which percentage of the nucleotides are complementary to this gene. And this should, of course, also be 100%. Again, because you specifically designed the primer to bind to this sequence. And here, luckily, as expected at the top, we find Homo sapiens, so human tumor protein p53, as the gene that the primer will bind to. And you can see that in this case, it's transcript variant 14, but it will also be able to bind to the other transcript variants of the gene. So transcript variant 10, 9, 12, 13, and so on. So normally you will be able to find all the different transcript variants of the gene here, unless you designed primers in such way that they will specifically bind to one of the transcript variants. So in that case, you should find first where the differences are between different transcript variants and find a part part of the sequence of a certain transcript variant that's specific only to that transcript variant. But as I mentioned, generally, when we want to measure the expression of a gene, we want to measure the expression of all transcript variants. And as you can see, this is the case for the primers we designed here. Now, what is very important to check is this so-called e-value. This e-value should be as small as possible because this represents the chance of finding a random sequence that matches equally good as the desired target. And here you can see that this chance is very low. So the chance of finding a random sequence that binds equally as good is very low. So this means that our primer is specific to this gene. When we now scroll down in the list, you will see that the next hit in this list, which is a gene different from p53, has a much higher e-value. So here the e-value is 38. And ideally, the difference in e-value should be 1,000-fold. So if here you find a value of 0.010, then the e-value of the next hit, which is a different gene than the one that you designed the primers for, should be, in this case, at least 10. You can see that it's 38, so it's even higher than 10. So in this case, it's perfect. So I did this now for the forward primer, but I should now do the same thing for the reverse primer. So I will do this as well. I will copy the sequence of the reverse primer. Here it doesn't matter whether you take the reverse complement or not, because it will search on both strands anyway. So we're now also blasting the sequence of the reverse primer. Voila. Then we will again see that the reverse primer will bind to the different versions of, or the different transcript variants of p53. But now we will see that the next hit, which is a different gene, also has a relatively low e-value. So in this case, the e-value is 0.16, whereas ideally we would want it to be 10 or higher. So normally in this case, what I would do is go back to my primer tree output and look for another potential primer pair in the output list. So in this output list, I would go to another potential primer pair, again, check whether it binds around or on top of an exon-exon boundary. And in case it does, I would again blast these primers. And then after checking several primer pairs, I would select the best or the most suited primer pair. So a primer pair where one of the primers binds on top of an exon-exon boundary, or where the two primers bind around an exon-exon boundary, and where the blast yields suitable results, which means a large difference in e-value between the gene of interest and the next gene in the list. And in this way, you can design primers for different genes of interest and also for potential reference genes. After watching this demonstration, I hope that you will now be able to use this knowledge to try and develop primers yourself for genes of interest that you want to measure using qPCR.